It may seem a bit surprising, but many animals can develop normal female individuals without receiving sperm from males. This is called parthenogenesis. However, in mammals, some genes on both females and males are suppressed. Therefore, genomes from both sexes are essential to have one set of expression-capable genome. So, in mammals, parthenogenesis is impossible. Parthenogenesis is a reproductive method where females produce new female individuals, without fertilization by sperm. Advantages of this system include saving energy and time by eliminating the need to find a mating partner, allowing for rapid population growth. Disadvantages include lower genetic diversity between parents and offspring and among offspring, potentially leading to decreased adaptability to environmental changes. Parthenogenesis in triploid carp, Ginbuna. Most triploid carp individuals are triploid females. The 3N oogonia undergo special meiosis to produce 3N eggs. The eggs are activated by sperm from related 2N species, but the sperm nucleus is excluded. This results in female offspring with the same genetic composition as the female parent. Therefore, triploid carp increase their population through parthenogenesis, and the relationship between individuals is clonal. Honeybee life cycle including parthenogenesis. In honeybees, only the 2N queen bee has reproductive ability and lays 1N eggs. If these eggs develop without fertilization, they become 1N males. So, honeybee males are born through parthenogenesis. 1N males can form 1N sperm without meiosis during spermatogenesis. Eggs are fertilized by sperm, resulting in 2N individuals. All 2N individuals are female. Female larvae that develop without royal jelly become 2N worker bees without reproductive ability. AAA, only one female given royal jelly, becomes a queen bee, with reproductive ability, and can produce eggs. Parthenogenesis by fusion of egg and polar body, automixis. In addition to clonal reproduction in triploid carp, some fish and reptiles exhibit automixis, where the egg fuses with a polar body to form a 2N egg that develops into a female individual. In this case, the chromosome combinations in the oocyte change, so the female parent and female offspring are not clones. Therefore, some genetic diversity is maintained among offspring. This type of parthenogenesis allows reproduction without males, but automixis has the drawback of limited genetic diversity in offspring. Organisms where parthenogenesis is blocked. Parthenogenesis is not possible in all organisms. Marsupials like kangaroos and placental mammals have genetic systems that block it. In marsupials and placental mammals, some genes on the autosomes of both females and males are pre-suppressed. This is called genomic imprinting. Therefore, genomes from both sexes are essential to have one set of expressible genome. Genomic imprinting blocking parthenogenesis. The phenomenon where only one parental allele is transcribed on homologous chromosomes is called genomic imprinting. Genes transcribed only from the paternal genome are called paternally expressed genes, PEG, while those transcribed only from the maternal genome are called maternally expressed genes, MEG. Genomic imprinting through DNA methylation. In marsupials with underdeveloped placentas, about 60 genes are estimated to undergo genomic imprinting. In developed placental mammals, 100 to 150 genes are estimated to be imprinted. Such gene suppression in somatic cells is maintained throughout life through DNA methylation and histone modifications. It is estimated that such genomic imprinting does not exist in monotremes, which do not have a placenta and lay eggs. Therefore, in marsupials and placental mammals, even if a 2N egg formed by fusion of an egg and polar body, where both genomes are maternal. So, it results in the absence of paternally expressed genes. This causes embryonic development to stop early in pregnancy. Chromosomal distribution of PEGs and MEGs. Paternally expressed genes and maternally expressed genes form clusters, concentrated in specific parts of certain chromosomes. This allows for efficient application of epigenetic control mechanisms, such as DNA methylation and histone modification. Reimprinting. Expression suppression is maintained throughout life, in somatic cells, due to epigenetic mechanisms, like DNA cytosine methylation and histone modification. However, in sperm and eggs produced by the next generation, PEGs and MEGs must be reset and reprogrammed to suit each sex. In fetal primordial germ cells, DNA demethylation, necessary for PEGs and MEGs reprogramming, occurs at a gender-specific time. Sex-specific reprogramming methylation occurs after days 15 to 16 post-fertilization in males and 2 to 3 days after birth in females. Role of genomic imprinting, conflict hypothesis. Currently, there is no definitive explanation for the role of genomic imprinting. An early hypothesis is the conflict hypothesis. This hypothesis states that in animals that exhibit viviparous reproduction, evolutionary pressures act differently. Males evolve to make their offspring larger, while females evolve to make their offspring smaller to reduce. This leads to the hypothesis that paternally expressed genes promote fetal growth, while maternally expressed genes suppress growth. However, as research progressed, and more imprinted genes were discovered, exceptions to the conflict hypothesis have increased. Typical roles of PEGs include involvement in placenta formation and maintenance. Typical roles of MEGs include suppressing fetal growth, regulating metabolic processes in fetuses and newborns, and contributing to the development and normal formation of the nervous system and brain. The content of this video can be viewed in text and images on the home page site, biology website, Life Science Lectures for You. The site address is given in the description field of this video.
I now conclude this lecture.
If you have not found it yet, keep looking.